everyone. Joining us today is Dr. Lydia Contreras, professor at the University of Texas at Austin's Department of Chemical Engineering. Dr. Contreras, would you like to tell us a bit more about your background in research?
Sure. So it's nice to talk with you all and be here. So my lab works on a part of chemical engineering that it's really devoted to creating applications, a more fundamental understanding for biotechnology. So this whole discipline of biotechnology really arises from the desire to use biological systems, so bacterial cells, human cells, eukaryotic cells, plant cells, as factories. So little entities or systems that can be controlled very deliberately to make any molecules we desire and we want. And this is particularly exciting when you can think that those molecules can have commercial values. So these molecules can be used for pharmaceutics, as precursors to make fuels. They can be used as polymers, as chemicals, etc. So this is really what continues to boost this interest in making molecules cheaper, but also making them in a much more green or environmentally conscious way. So the field of synthetic biology has really progressed quite a lot. And it's been really exciting that now we can synthesize DNA. This is pretty cheap to do. So we can actually synthesize base by base, put DNA together. And once you put DNA together, it becomes really a game of putting it in cells and have cells read that message to produce anything that you want them to produce. So this has quietly expanded a lot our dreams when it comes to synthetic biology and how far it can take us. So for example, if you think about the fact that we can build these and engineer units that behave like living systems, now what you're talking about is just not DNA being synthetic, but entire systems of biology being synthesized so that you can put them in cells and cue them to deliver drugs on demand, or so that you can put them in bodies, in human bodies of patients and hunt down specific cells like cancer cells, or detect toxins in chemicals that are harmful and send us signals or become diagnostic, etc. So we're really interested in this field, and we're really interested in sort of engineering these regulatory pieces of cells. So really controlling how cells sense their environment and how they respond. So there's a lot of engineering here. There's a lot of dynamics. There's a lot of coordination with the environment. There's a lot of trial and error and engineering parts together. And particularly what we look at is what you all have been learning in biology, which is this idea that you can put DNA goes and makes a transcript, an mRNA, and then you can make a protein. So what my lab really focuses on is in engineering these pathways so that we can now put synthetic DNA and make synthetic RNAs and direct so that we can make proteins that are not naturally encoded in bacteria, but that we really want to make. So they can be foreign proteins again, or foreign chemicals or foreign pathways, biomolecular pathways that have molecules of interest and economic value. And particularly we focus on these RNAs that do not encode for proteins typically, but what they do is really regulate this entire machinery based on external stimuli. So I just want to skip ahead and tell you lastly that there's a lot of tools that we use. This research is very interdisciplinary, as it is a lot of the research in synthetic biology and in biochemical engineering. So we use a lot of these concepts of synthesizing pieces. So there's a lot of design that goes in there. Designing robust parts that you can throw in a cell and the cell won't chew it up, it won't degrade it. It will be stable, it will be specific to what you want to target. So there is a remarkable amount of molecular design that goes in it. There's also quite a lot of emphasis on bioinformatics. So this is now using a lot of mathematical training and computerized skills to go and search for what's already there and to compare lots and lots of data and find patterns. So this is what we call machine learning patterns that can better inform us to design these parts. There's a lot of biochemistry and genetics that we use to characterize these in cells. And using these, we also make a lot of models. So there's a lot of theories that go into how these parts function. They function together in cells, etc. So we can actually make mathematical models. So in essence, this is a great area. It's one of the many that an engineer, a chemical engineer, it's active in participating, but there's also electrical engineers involved and computer engineers involved, and people that are really interested in designing systems that would operate and would function the way you design them and then characterizing them. So a lot of different disciplines involved. Usually these are teams that involve a lot of scientists, engineering, and so it's quite a complex but very interesting research area. So I'll stop there.
I just had a couple of questions for you.
Yep.
I just wanted to start off with what sparked your interest in synthetic biology?
Yeah, that's really a great question, Karthik. So when I was growing up, I still remember I had a lot of interest in medicine. I wanted to be a doctor, a medical doctor. And I think that part of that is because that's what I knew. You grew up going to the hospital, seeing doctors, seeing what they did. When you thought about engineering, my vision of an engineering, the only type of engineering that I knew at the time was civil engineers, because I could see bridges, right? I could see bridges, or mechanical engineers. I can see plane designs. I heard of NASA and going to space and what they did to design those space shuttles, etc. But I really became fascinated in high school. When I was going in high school, this is where the Genome Project was going on. So people were really, for the first time, discovering all the genetic sequences that made up a human being. And it was fascinating. And I remember being caught up in this idea that if we know what makes a human being, we can actually synthesize things. We can synthesize biology to do really powerful things in society. So I remember that, and I became really interested in that concept very early on. So I pursue a lot of science in high school. I pursue a lot of AP math, AP chemistry, AP biology, and started learning more and more. And I was pretty interested in sort of the huge potential for this field to integrate engineering, but also biology. So I turned away a lot of my attention on medicine from a practitioner's perspective, from a physician that would practice medicine, to one that would think of applications that can be engineered to make the medical field much more effective for every human being. So I became really interested in the power of synthetic biology as applied to the medical field.
Got it. Yeah. And what have you learned about how cells react to synthesize DNA?
Well, so the ability to now synthesize DNA and throw them in cells, that's actually pretty sort of well figured out for model bacteria. But funny you ask, because they're still trying to, protocols being trying to develop to actually effective ways that bacteria, especially those that we don't know anything about, to uptake that DNA. Right. So there's several methods that have been designed for bacteria to actually uptake it. You have to weaken their membranes by various tricks, electrical signals, or weakening with chemicals, right? So that their membranes kind of rupture just a little to let the DNA in. And then the trick is incorporating that DNA, reading it and treating it as really a command, so that the cell can actually obey the signals, right? Or follow the signals that you're putting in there, encoded in that piece of DNA. I think the new frontier really is to figure out how to do that in bacteria that we don't know anything at all. And believe it or not, there are so many diverse species of bacteria that we still have no idea. Even bacteria, right? You think, oh, bacteria, the simplest organisms. So when it comes to bacteria, when it comes to having them even in communities that are made up of different bacteria, hundreds, thousands of them, it's going to become really interesting to think about putting in those DNA signals or DNA instructions, right? Genetic instructions into bacteria to reprogram them. So that field is still evolving, just getting more complex.
Okay. Yeah, that sounds really interesting too. Just the next question we had was, what challenges did you face when dealing with RNA proteins?
Well, that's another really good question. So one of the things that, the joke of people that work with RNA is they'll tell you, don't try to save you, right? Don't protect yourself from the RNA, right? You always think of, protecting yourself from the lab, from experiments. They tell you protect the RNA from you, because RNA can actually be experimentally difficult to work with, in terms of it could be unstable. You have to kind of keep it cold. You can have any enzymes or any proteins around that would degrade it. And so the half-life can be short, meaning that it can be, the stability can be poor. So there's a lot of challenges with working with RNA. And of course, it's a very unknown still frontier in science. So kind of, what makes RNA starts to be producing cells, what cues that, and how is that integrating with environmental signals, like the environment changes. All of a sudden, bacteria experiences pH when it comes to your gut. What signals it to start all that central dogma that we talked about? DNA going to RNA, RNA making proteins. What signals that machine to go or to stop if it's under too much stress, and it just shouldn't waste all this energy trying to make unnecessary molecules. So there's a lot of those questions of the signaling, the regulation, and even more interesting, the fact that, when I was in high school, I learned that RNA was made up of A, C, Gs, and Us, right? That's what you learn. But now we know that every one of those bases, A, C, G, U, can undergo significant chemical modifications, hundreds of them. So there's all these little variants that you can actually make RNAs like puzzle pieces. And you have many puzzle pieces. And so when you mix and match, what do you make? What do all those chemical diversity, what does it mean for RNA function? So there's still a lot of challenges with really even understanding RNA, let alone programming and synthesizing it in ways that are efficient and controllable.
Okay, yeah. And just our next question for you would be, like, what do you envision the future of chemical engineering to be?
Pretty broad. So the great thing about chemical engineering, it's that it's a discipline that trains you very widely to really think about how systems work, right? And so classic classes in chemical engineering are thermodynamics, so energetics. So a lot of the models that I even showed you earlier, they had delta G terms, they had free energy, because we think about energy a lot in terms of what are the forces that are driving these molecules, right, to fold or to assume structures that are stable, right? So energetics is something that applies all the way from biology to the brain to any other challenges that are out there with, clean energy sources, solar, wind, etc. So the other areas of chemical engineering are transport. So how things actually, what gradients transport anything around, molecules, energy, etc. So those are wide concepts, right, that apply to modeling, that apply to industrial processes. There's another thing in chemical engineering area of optimization. So optimizing processes when it comes to materials and times and energy, that's key. From traffic to banks to, anything, right? Classes scheduling, optimizing that to what I do, right? How these enzymes optimize each other, working together, etc. So my point is that chemical engineering classes are very fundamental in terms of describing processes. And since you pretty much can describe anything as a process, applications of chemical engineering are only beginning to grow. So this is one, but I have colleagues that work on energy. So designing better energies on batteries, right? More efficient, smaller batteries. I have colleagues that work on materials. So, right, materials that are from chips in your computers to materials on windows that better capture sun energy and store it. I have colleagues that work for the car industry in paint shops, really designing processes that lower toxic emissions to the air, right? So environmental type of applications to chemical engineering. So there is really a wide range of applications. And as you know, there are major world challenges, right, in environmental engineering with accessing clean water, accessing clean energy sources, from medical issues like personalizing medicine so that we can actually give somebody targeted medicine with less side effects, etc. To understanding the brain. How does the brain work and how can we reprogram it? How can we recreate vision so that people that are not naturally born to see can see. So there's all of these challenges that are out there where chemical engineers are playing major roles.
Yeah, got it. And just what field do you think would be most impacted by future discoveries in synthetic biology?
There's a few. So agriculture is one to have expected some major impacts, right? How we think of plants, how we think of creating plant communities synthetically where products, can be more efficiently harvested. And as we know, food and access to food is a major issue around the world. Something we don't think a lot about as Americans being in the United States, but around the world, access to food for everyone is a major issue. So agriculture is one. As I mentioned, over my talk, medicine, of course, right? That idea of targeting medicine and delivering medicine, specialized, targeted, personalized to specific people in a very kind of, individualized way. It's another one. Also expected. So yeah, medicine, agriculture. Those are two of the ones that come immediately to the top of my head. Right? Products in terms of anything that we make. So safer and more environmentally conscious products, right? Where we might not have to be using traditional resources that might not be as environmentally friendly. So definitely environmentally conscious products. I think it's another one that has great potential.
And our final question for you is, how can high schoolers dive deeper into biomolecular engineering?
Well, that's one of the things that I suggest is you guys can do, book clubs or paper clubs, just like you're doing now, where you actually follow a couple of people and talk about different review papers are pretty good to read. So there's different types of papers that academic people like me publish, right? Some of them are very heavy and very hard to read. They have a lot of data and experiments, and they require a lot more understanding of the background of those research areas. But there's other ones called reviews or book chapters that really provide an overview of the field. So like you can go and look up for reviews on synthetic biology or current opinion papers, current opinions that kind of more broadly describe how that area fits in for students to kind of dive in. So those are great things to discuss with each other and invite a professor like me to come and talk about these areas with you all. So that's one. Number two, of course, keeping up with your AP courses if you have the chance, or with your science courses in school and math. Other things that you can also do are to, as soon as you can, especially as you start approaching your junior, senior years, seek for opportunities to work in a lab over the summer, volunteer, learn, be around people doing research. So those are ways that have been very effective for some of the high schoolers that we've hosted in our lab to learn more hands-on, right? Hands-on of what's going on.
Well, everyone, thank you so much for watching our interview with Dr. Contreras. Take care, and we'll see you all next time. Bye.